All right, good morning. It is time for our last two labs. It's going to cover the nervous system and then also sense organs. So, sense organs shouldn't take very long. I think we're just going to cover the eyes and the ears. We'll quickly go through the lab, and I'll give you just enough that we need to be prepared for the test. So if we go through the nervous system lab, we can begin with these objectives. First thing we're going to do is we're going to look at the neurons. Then we're going to look at the brain. We're going to identify some of the parts of the brain. We'll talk briefly about some of the functions of those parts. We'll look at the spinal cord, specifically looking at a cross-section of the spinal cord, and we'll talk about some functions of that. And speaking of the spinal cord, there is a process called the reflex arc that we will look at that as well. All right, so in the introduction, just some information that the nervous system is made up of what's called the central nervous system, and that includes everything right down the middle. So that's the brain and the spinal cord. Everything else includes all the nerves of the body, and the nerves extend out from the brain and spinal cord. We would refer to those nerves as being part of the peripheral nervous system. We have different types of nerves. We have sensory nerves that are composed of sensory neurons. What they do is they detect changes from the outside environment, and they bring them in towards the spinal cord and brain. We also have motor neurons that send signals the opposite way. They begin with the brain and spinal cord and go out to what are called effectors. In this case, effectors are going to be muscles or glands, right? All right, and the basic unit of structure and function for the nervous system is going to be the cell, right? That carries signals. These cells are called neurons. All right. So the nerves carry information as electrical signals, also referred to as action potentials, through the entire nervous system. And we can divide these neurons into different parts, including dendrites, cell bodies, axons, and synapse. And we'll show you what those look like in just a minute. Just keep in mind, this is kind of important, that when we look at the tissues of the nervous system, there's two major types of tissues. There's gray matter, and then there's white matter. All right. So if we're looking at gray matter, pretty much we're looking at everything that's in the cell body, right? So that includes collections of cell bodies. So that's going to be the nucleus, the cell body, and then the dendrites. So coming off of a neuron is a really long section, which is called the axon. And the axon is covered with a fatty substance called myelin, and it appears white. Okay, the myelinated axons appear white, and they appear different from the gray matter. They look different from the nucleus. The nucleus is darker, so that tissue appears gray. The axons are lighter, that appears more white. And it's very easy to distinguish the difference between these tissues when we're looking at the brain. You could clearly see there's white matter and there's gray matter. And when we look at the spinal cord, you could see that there's gray matter, which is separate from the white matter. And I'll show you what that looks like when we get to it. So just remember, if you remember the white matter is made up of axons, then you remember the gray matter is pretty much everything else. All right, so this is a picture of a neuron model that we would normally have at our desks, right? And so let's start by labeling this. The first thing that I think we should label that makes sense is this thing right here, this ball right in the middle. That's going to be the nucleus. All right, right there. And so what holds the nucleus is the cell, right? The cell holds the nucleus. So this cell actually is referred to as the cell body. This arrow right here is pointing to the container that holds the nucleus. So let's write cell body. That one. Of course. This thing gets me every time. All right. Then you can see there's a long extension coming off the cell body. Actually, this extension has been cut right here. They cut it here, right? And it continues here because it's too long to fit on the page. But this piece down here, this section down here would be continuing over here. So this right here is actually the axon. So let's label this the axon. And this is also the axon. This is a continuation of the axon down here. All right. The very beginning of the axon is kind of an important part of the neuron. This area right here, the beginning of the axon, where it extends from the cell body. This is called the axon hillock. And this is where impulse is generated, right? So the cell, if it wants to fire a signal, it collects smaller signals from the cell body. And once it reaches a threshold, the axon hillock is the area that generates the action potential down the entire axon. If we get a closer look at the axon, we can see that there's these specialized cells that are wrapped around it. These cells have a cell membrane that is made of lipids, right? Like all cell membranes. And as it wraps around the axons again and again and again, it creates a nice fatty insulator. So these cells that are doing this, these are called Schwann cells. They produce what's called the myelin sheath. And the myelin sheath acts as an insulator. And what that does is that protects the current, right? Much like the insulator on a cord. Here, I have a cord here. There's electricity running through this cord, but the electricity isn't lost when I touch the cord because of this rubber insulator. Your cells have an insulator. Your neurons have an insulator. It comes in the form of fatty substance called myelin. And that just kind of ensures that the signal continues without being interrupted. There are some sections on the axon where we don't have the insulator, right? And these produce these little nodes. These nodes are called the nodes of Ranvier. Right? And these nodes are important because it's in this little section that ions could be exchanged between the inner cell environment of the neuron and the extracellular environment. And this is how we conduct the electrical current. So if the ions are only permitted to be exchanged across the cell membrane at these nodes, what this does is this creates a faster signal. So myelinated axons tend to be faster because the ions are being exchanged only between the nodes. So we have this kind of like electrical hopping that goes all the way down the neuron. This hopping is referred to as saltatory conduction. If we go back to the cell body, we can see that there's these little hair-like things there, or these little branches, I should say, that extend off of the cell body. And what they're doing is they're looking for signals. They're bringing signals in, all right? These are called dendrites. Dendrites, all right? But you can see the cell body and the dendrites are, it looks like they're touching these other structures here, but they're actually not touching, but they are very close to. These are the ends of the axons of another neuron. So these are called axon terminals. These axon terminals are not part of this neuron. They're part of a neighboring neuron. So a neuron that maybe wants to communicate with this neuron. All right. I think we got them all. So how could we describe these parts? Well, we could say the dendrites are branches that bring information into the cell body. They carry what are called really small signals, right? These really small signals are called graded potentials, right? So if the dendrites are bringing in graded potentials, which are small signals, into the cell body, if it reaches a certain level, which we call the threshold, if it reaches the threshold, then that cell body will generate a brand new impulse and shoot it down the cell, the axon. So the cell body is the area that contains nucleus. Axon hillock is the beginning, the axon, and this generates an action potential. Schwann cells, what they do is they wrap around axons and they produce a myelin sheath, which acts as an insulator. The nodes of Ranvier, these are the areas on the axon that are not covered with myelin. So this is where the ions are being exchanged between the inner portions of the neuron and the outer portions of the neuron. So this is where we're going to call this the site saltatory conduction. All right. At the very end, there's a space between the axon terminals. The axon terminal is the end of an axon. And the space between the neuron and the effector is called the synaptic cleft. All right, so remember, the neurons don't actually touch the effectors. Remember, the effectors could be, let's say, a muscle. Let's say I want to send a signal from my brain to my muscle. Well, the signal would generate in my brain. It would then get transferred to the spinal cord, right? So this is a motor neuron. From the spinal cord, the motor neuron sends the signal, one neuron, to the muscles in my arm, but the neurons don't touch the muscles of the arm. They don't touch. There's a space between, and it's between that space that the neuron would have to send a chemical signal in order to influence the contraction of the muscle. And that space is called the synapse or synaptic cleft. So what is the function of neurons? Send signals. Signals are not only electrical signals, right? They're electrochemical signals. Where are the neuron cell bodies located in the brain or spinal cord? Well, in the very beginning, we said the white matter was only axons. So it's going to be, in this case, the cell bodies is going to be gray matter, right? Where are the axons? Well, the axons are going to be white matter. So let's do that. What part of the neuron is covered with myelin? Well, of course, that's the axon. And name the cells that form the myelin sheath. Well, these are the Schwann cells. And what is the function of the myelin sheath? That is to insulate axons. Remember, myelinated axons send signals faster than non-myelinated axons. There are non-myelinated axons in the body as well. All right, so if we look at a neural smear, let's go to the lab atlas for that. We have a neural smear. Here it is. This is a picture of a neuron, a microscopic observation, and this is a high power view, and you could see the dendrites are these branches. All right, the cell body is the area that holds the nucleus. There's the nucleus. And we have one long section called the axon. All right, so this is going to be your microscopic observation. All right, so try to draw those parts. And label these parts too, please, and fill in this information as well. All right, if we look at the brain, we could do some labeling here. We could start at the top and work our way around. At the top here, we have what's called the cerebrum. And then we have this other section here. The cerebrum contains all the major lobes, right? So we have the frontal lobe, we have the parietal lobes, we have the temporal lobes over here on the side, the occipital lobes. And the cerebrum is kind of controls all the things that you have control over, right? Like your planning, your thinking, your movements. So all voluntary actions are controlled by the cerebrum. We do have a right and left hemisphere in the brain. Typically, the right side of the brain controls the left side of the body. The left side of the brain controls the right side of the body. But these two sides need to communicate with each other. And this is where the corpus callosum comes in. Corpus callosum is one of the most easily recognizable structures in the brain. If you cut the brain in half and look at it like what this model is showing us, this is we're looking at the right hemisphere having the left hemisphere removed. So we're looking at a medial view here. Inside here, we have what's called the thalamus. Thalamus. The thalamus has this structure underneath it, and we call this the hypothalamus. Hypothalamus we've talked about a lot because this is the control center for homeostasis. The thalamus is more like a relay station of the brain. So all incoming information from sense organs is coming up the spinal cord. It gets relayed through the thalamus. So the thalamus is much like the grand central station where information is coming in. It goes through the thalamus, and then the thalamus dictates what lobe it needs to go to. This thing down here is the pituitary gland. Pituitary gland is the master gland for the endocrine system. And the pituitary gland produces many hormones that have the ability to either turn on or turn off many of the other glands of the body. Over here we have the cerebellum. Cerebellum is responsible for the body's ability to conduct smooth muscle movements, as well as balance and coordination. In the brainstem here, we have this area up here. This is called the pons. The pons plays an important role in respiratory rates, maintaining respiratory rates. And then down here, the lower part of the brainstem is called the medulla. And the medulla is the center for involuntary actions. So it controls those things that you don't really need to control. Things like heart rate, digestion, all those other things. And then the brainstem leads to the spinal cord down here. All right. So if we look at the sheep brain, which we would be able to, you could very easily see the cerebrum at the top, the cerebellum, and then the brain stem. You would also be able to see that there's this tissue that surrounds the entire brain. There's actually three of them, but these include what are called the meninges. Meninges. All right, so this is a collection of protective tissues that cover the brain and the spinal cord. And these produce fluids, right? The fluids, the cerebrospinal fluids that also provide protection. All right, so what could we say here? The right hemisphere controls outside of the body, and the left hemisphere controls the right side of the body. Corpus callosum connects the two hemispheres, right? This is a bridge inside the brain. All right, when we look at the different lobes of the brain, I mean, there's a lot of functions there, too many to fit in here. But the frontal lobes are important for decision making. Oh my goodness. This thing does this to me every time. Planning. That kind of stuff. Thinking, emotions, your behavior. This is a big part of your personality is coming from your frontal lobes. Moving on. Let me see. I should go back in there. Is that going to work? The temporal lobes. This is for memory. A lot of memories. Language, hearing, especially. Big part for hearing. The parietal lobes. Classification. Knowledge of numbers, spelling, things like that. The occipital lobe is for vision, right? This is your primary visual cortex, right? So that's all the way in the back of the brain. But also the processing of vision, processing of colors, that kind of stuff. The thalamus is a relay station. I just do that. Information. The hypothalamus is the site homeostasis. Right? And the pituitary gland is, we're going to say that this is the gland that this is the master gland. Endocrine system. The cerebellum. Unbelievable. The cerebellum helps the body to maintain balance and coordination. The pons is part of the brainstem. And what the pons does is it helps to control respiratory rates. Medulla controls involuntary actions of the body. So all those things that are out of your control. Digestion, blood pressure, things like that. Olfactory is for smell. And this is smell is a sensory function, not a motor function. So what we're going to do is I guess we could circle sensory there. Optic is another sensory function. Oculomotor, however, is a motor function. And with optic, these are cranial nerves. So these are coming from the brain. The optic nerve is the second cranial nerve. This is for vision, right? So this is taking information. Oh my goodness. From the eyes and then directing it towards the occipital lobe of the brain. However, the oculomotor nerves of the brain are also going to the eyes, and what they're doing is they're controlling the muscles of the eyes. All right. And so we have ventricles. There's 4 cavities inside the brain that help to produce cerebrospinal fluid. And then we have the meninges. The outer meninge is called the dura mater. The one beneath that is called the arachnoid mater. And the one beneath that is called the pia mater. The pia mater is the one that's actually in direct contact with the brain. But the cerebrospinal fluids is produced within the ventricles, but then it moves from the ventricles to the outer parts of the brain all the way down around the spinal cord. All right, so this is a sheep brain. A sheep brain would have been provided to us in the lab. And you could see the olfactory nerves very easily. You could see the optic nerves very easily. You could see the optic nerves branch in 2 different directions because we would have 2 eyes up here. But you notice if you follow the optic nerve, they crisscross here. This area right here is actually where they crisscross. It's called the optic chiasma. And then these nerves would continue all the way to the back of the cerebrum where the occipital lobe is, because it's all the way in the back at the occipital lobe that we have the primary visual cortex. So we're looking at the brain from underneath. Right behind the optic chiasma, you would very easily see the pituitary gland. On the sides, you would find the temporal lobes. Right behind the pituitary gland, you would have the 2 oculomotor nerves. And then we would get to the brainstem. From the brainstem, the very top of the brainstem is called the midbrain. But then we would find the pons and then the medulla, finally leading us to the spinal cord. All right, so this is looking from the side. This is a lateral view here. We have the corpus callosum. The corpus callosum is this structure right here. Very easy to find. Looks like a big question mark. The hypothalamus would sit out front over here. The thalamus would be behind it over here. The pituitary gland would dangle like this out in the very front. Again, the brain stem has three parts: the midbrain, the pons has a nice barrier around it, and then the medulla. This is all brain stem right here. So remember, the brain stem controls all the involuntary functions. The cerebellum is very easily recognizable because it has this white matter here that branches. It looks like a tree. In fact, this white matter inside of the cerebellum is called the arbor vitae. And arbor vitae literally means tree of life. All right. I'm going to let you guys do this on your own. So this is just a little bit of research. What you're going to do is you're going to put the part of the brain that is responsible for these functions. All right. Some of them we kind of already did. So it won't be too hard. If we look at the spinal cord, the spinal cord begins at the very base of the skull. It's really just a continuation of the brainstem as it leaves the hole on the bottom of the skull called the foramen magnum. It extends all the way down your back and keeps going till about the L1 or L2 vertebrae. So it's about 40 to 50 centimeters long, about 1 to 2 centimeters in diameter. And again, just like in the brain, there's going to be white matter, there's going to be gray matter. We could divide, remember how we divided the vertebrae into sections? These were the cervical vertebrae here, right? The bones we're talking about now. So it begins with atlas and axis, C1, C2, all the way going from C1 to C7, right? These are the bones. And then we get to the thoracic vertebrae, going from T1 to T12. Finally to the lumbar, which is L1 to L5. So our spinal cord would begin at the foramen magnum, go all the way down here through all the cervical vertebrae, through all of the the thoracic vertebrae, and go through somewhere between L1 and L2 where it would stop. However, there's still nervous tissue continuing through here. You can see the nerves. The nerves continue down even through the sacrum. So this is the sacral vertebrae, S1 through S5. These are fused together, so it looks like just one big piece here. So sensory nerves, remember there's different types of nerves. There's nerves that are afferent and there's nerves that are efferent. The afferent nerves bring things in. So sensory nerves carry signals from the sense organs to the brain. Where the motor nerves carry signals from the brain to the effectors. The effectors are either muscles or glands, and I'm not going to write that there because that's our next question. And mixed nerves, what do they have? Well, they have both, right? Mixed nerves are nerves that contain both sensory and motor neurons inside of them. So they carry signals both ways. We could organize these nerves into major networks of where they intersect with each other, and then we could kind of categorize these into four major categories. We have a cervical plexus, the brachial plexus, lumbar plexus, and the sacral plexus. So we could kind of identify these on the model here. Well, first of all, this is the brain. Let's label the brain. And this is the spinal cord. All the yellow lines in here are going to be nerves. Not me again. And then we have the four major nerve plexus. Okay, in the neck, number one, that's going to be our cervical plexus. Anything that goes towards the arms, the word brachial is used a lot, so we could see this collection of nerves extending down into the arms. We're going to call that the brachial plexus. And then we have the lumbar plexus, and then the sacral plexus, right, is number four. So it was cervical, brachial, lumbar, sacral, with one C. All right. Gray matter. Oh my goodness. I don't know why that keeps doing that. Anyway, the gray matter contains the cell bodies. And the nuclei. The nuclei are darker. That's why this tissue appears gray. White matter contains myelin. Myelin is a light-colored fatty substance, so this contains the myelinated axons. The central canal is in the middle. This contains fluid. If we're looking at a slice of the spinal cord now, the dorsal root is the site to say it contains actually sensory nerves. The dorsal root ganglia is a collection cell bodies the sensory nerves. The ventral root contains motor nerves. And spinal nerves. These are mixed nerves, so they contain both, and I'll show you exactly what we're talking about for all of these things here. All right, again, so if we look here, this is a slice of the spinal cord, and it looks like we have this butterfly-looking structure in the middle of the spinal cord. You can see that this color is darker than the surrounding color. Well, this is the gray matter. This is where all the cell bodies that contain the nuclei are found. This is where they kind of hang out. Everything out here surrounding that this is all white matter. If you look at the spinal cord, the gray matter is in the middle. The white matter is on the outside. It actually is the opposite in the brain. In the cerebral cortex, the gray matter is on the outside, and the white matter is more on the inside. But anyway, the spinal cord, it sends and receives signals. So when signals are coming in, they're coming through this mixed nerve here, this spinal nerve. And then when they get to this intersection, they always go this way. This is the dorsal root. So this is the back side. This is the front side. This area, this would be the front side of your body. So sensory information comes into the backside. This is the sensory neuron right here, right? This is the dorsal root. Let's label that. Oh my goodness. You see, when you look at the dorsal root, get a closer look at it here. It has this bulge over here. If we look at the other side, it bulges over here. This is where the sensory neuron cell bodies are conglomerating, and it creates this bulge. Anyway, this bulge has a name. This is called the dorsal root ganglia. Oh my goodness.
Is that always scheduled? Oh, daytime, jeez.
That's the dorsal root ganglia. Down below here, this is, if we're sending information out, this is the efferent pathway. It would come from the spinal cord and it would go the other way, right? So the outward information leaves the spinal cord on the ventral side. So this would be a motor neuron. The motor neurons kind of meet up with the sensory neuron inside the spinal nerve. This spinal nerve, that's why they call it a mixed nerve, because it has both types of neurons. So it's afferent and efferent, right? So this is the ventral root. I'm just going to abbreviate. You write in ventral root right there. This is the mixed spinal nerve.
Thank you.
All right. Here's our central canal. The central canal is right in the middle, and this would contain spinal fluids. Central canal. It's a lot of fun writing with the mouse. And then, of course, we have white matter here and gray matter there. All right. The spinal cord is involved in what are called spinal reflexes. All right. So a reflex is a really fast response to something, and the speed of it is important in order to minimize damage, right? So let's say that you get stuck with a tack here. Well, you have specialized cells in your skin that would, you have the receptors in the skin that would pick up the stimulus. These receptors that would then pass the stimulus onto a nerve like this one. All right, so here we have the stimulus. This gets passed on to a very specific kind of nerve that brings information in. This is a sensory nerve. The sensory nerve, you can see, has that bulge there in the dorsal root. That's the dorsal root ganglion. Continues through the dorsal root into the spinal cord. Now we're inside the gray matter here. There's a third type of neuron that is called an interneuron that would take that information and immediately pass it on to a motor neuron. The motor neuron would then generate a signal to the muscles, allowing you to quickly move your hand away from the tack. All right. Only after this response is generated will this spinal cord send that up to the brain for processing. And this is how we get quick responses. This is how we get reflexes. And the spinal cord handles a lot of reflexes. The brain doesn't even get involved. And this is the reason for that is to generate a response with the shortest amount of time in order to minimize damage. So from number one here, we could say that this is this is the stimulus. From the stimulus to the sensory organs. No, from the sensory neurons. I gotta type this because I can't write with my mouse. Sensory. Oh my goodness! Every time. All right, from sensory neurons inside the spinal cord, it goes to interneurons. Why does it do that? The interneurons to the motor neurons. And then from the motor neurons, it goes all up the ventral side. This is the ventral root. And then that mixed nerve, that spinal nerve, takes it all the way down to the muscles of your hand or wherever your arm. And of course, if it's a muscle, we would call that the effector. Right? And this whole process is called the reflex arc. Okay? So again, it begins with a stimulus. And that's the tack going in. It sends a signal onto a sensory neuron. It goes in through the dorsal side. From the sensory neuron, it gets passed to an interneuron. From the interneuron to a motor neuron, out the ventral side to the effector. And this is going to generate the response, right? Pull your hand away. If we were in class, we could demonstrate this. With hitting your knee with the hammer, like you do, like you've probably been through this many times at the doctor, where the doctor strikes the patellar ligament, and that causes a response, right? Because when your knee is closed, the nerve on the ligament is exposed, and when you hit the nerve, the spinal cord's response is to extend the knee so the patellar covers that ligament. So in this case, what is the stimulus? The strike. The hammer. What is the receptor? Well, it's pressure sensitive cells in the patellar ligament. All right, moving on. Which root has sensory neurons? That's the dorsal root. Where are the cell bodies of the sensory neuron located? Well, that's the dorsal root ganglia. Where's the integration center for spinal reflex? Well, the spinal reflexes are controlled by the spinal cord. Remember, the whole idea is we don't get the brain involved, and that creates a faster response. Which root has motor neurons? Well, the motor neurons come out the front side or the ventral side. So that's the ventral root. What is the effector organ for this reflex? Well, the effector organ, if you want to straighten or extend your knee, is going to be the quadriceps muscles. Right? That group of muscles. What is the response for this reflex? We want to extend the knee joint. This is a spinal reflex. And this reflex is used to test the integrity of the lumbar region. This is the lumbar region of the spinal cord. All right. This brings us to our next lab for sense organs. The sense organs lab takes a look at mostly the eyes and the ears. In the lecture, we get into the taste, smell, and touch. But pretty much for this lab, the big part of the lab is the eyeball dissection. So what I'm going to do is I'm going to attach a video of the eyeball dissection that I'd like you to watch. And we spent a lot of time looking at the models of the ear. So for this, I think I'm pretty much going to show you the video, label the parts of the eyeball, and then, well, they're labeled here already, so that's even better. Provide you with that model. And then what we can do is there's a bunch of these activities in here, obviously, that we're not going to be able to do. But what we can do here is I want you to label this diagram because this is kind of important. So this is the ear. So why don't we start with, we'll start from the outer ear. We'll go to the middle ear, and then we'll go to the inner ear. So the outer ear has this structure here. This is called the pinna. Also called the auricle. And what this does is it conducts the sound waves in. Into what's called the auditory canal. The sound waves are directed through the auditory canal into the middle ear. The middle ear begins with this structure right here. This is called the tympanic membrane. Tympanic membrane is also called the eardrum. So what happens is as the sound waves are coming in through the auditory canal, it causes this eardrum to vibrate. On the other side of the eardrum, we have these three bones, the malleus, the incus, and the stapes. Collectively, we call these bones the ossicles. And what the ossicles do is they vibrate. So what's happening is we're changing the form of the waves from sound waves to mechanical waves, right? As the tympanic membrane is vibrating, the vibrations are transferred to these three tiny bones. So we have sound waves getting transferred to vibrations, physical waves. The last of the three bones, the stapes, vibrates up against this structure in here. And what happens is this structure is filled with fluids. So we have another transfer from vibration, physical waves, to fluid waves. All right. So this area right here is not really for hearing. This area here all this is for balance and equilibrium. This is called the vestibule. And off the vestibule, you can see we have these 3 loops. These are called the semicircular canals. These are filled with fluid, as the vestibule is also. And when you move your body around, when you move your head around, these fluids shift, and it sends a signal down the vestibular nerve to the brain, and it gives you an idea of where your body's position is in space. Proprioception, communicating with the cerebellum, giving you a sense of balance. But the stapes here, as it's vibrating, it's producing fluid waves that go this section. We have this coiled section here that goes around and around and around. This is the organ for hearing, and this is called the cochlea. It's inside the cochlea that we have specialized cells that make up what's called the organ of Corti, that are affected by these fluid vibrations as they pass, and they send their own signal down the cochlear nerve to the temporal lobes of the brain, and this is how we process hearing. All right. So again, the sound waves get directed by the pinna into the auditory canal. They cause this tympanic membrane to vibrate. The vibrations get transferred from the malleus to the incus to the stapes. The stapes is banging up against this oval window here, which is the very beginning of the cochlea, causing fluid waves to transfer through this entire cochlea, around and around and around and around and around. And inside here, we have a structure called the organ of Corti that has very sensitive cells that are picking up these waves, sending signals to the brain, which are going to be processed as hearing. All right. This is the middle ear here. All this structure in between the cochlea and the tympanic membrane. This is middle ear. Everything on this side is outer ear, and everything on the other side of the stapes, so the cochlea, the vestibule, the semicircular canals, is all inner ear. And this is called the eustachian tube. This tube actually connects to the nasal cavities. And this is how we relieve the pressure in our ears. In fact, if you hold your nose and close your mouth and blow, you can feel your ears pop because this is connected to the nasal cavity. This is called the eustachian tube. Oh my goodness. All right. Okay. Now this is a closer look. So if we zoom in at this area right here, the structures of the inner ear, now we're in reverse. We have the cochlea, right, which goes around and around and around. We have our two nerves. We have the auditory nerve, which is also called the cochlear nerve. And then we have the vestibular nerve. The vestibular nerve kind of extends around the vestibule. Here's our vestibule here. The vestibule is divided into two sections. We have an utricle and a saccule. All right. Again, the utricle and the saccule have structures that communicate with the brain for balance. And in addition to that, we have these semicircular canals, right? Semicircular canals here. The utricle and saccule are more for gravitational equilibrium. So what does that mean? That kind of gives us information of like where our body's position is in space. It tells us if we're accelerating in a car. It tells us if we're going up or down in an elevator. You could feel that as these fluids are shifting. Where the semicircular canals is more being used for kinetic equilibrium or rotational equilibrium. It tells us if we're spinning around, that kind of stuff. All right. So if the structures are for hearing, or we could put an H. If they're for balance, we could put a B. If they're filled with air, put an A. If they're filled with fluids, put an F. Well, the hearing, this is for hearing, right? The auditory canal is for hearing. Tympanic membrane is for hearing. All these parts are for hearing, except for these two, really, the semicircular canals and the vestibule. These are for balance, right? These are filled with fluids. These are filled with fluids. The nerve is for hearing. The organ of Corti is for hearing. The cochlea is actually filled with fluids. It's for hearing. The organ of Corti is for hearing. This is the auditory nerve is not filled with fluid, so we could leave that blank. The organ of Corti is part of the cochlea, so we could say it's surrounded by fluids. The eustachian tube is for hearing, but it has air. The ossicles are part of the middle ear. They're surrounded with air. They're just transferring those vibrations from the eardrum to the cochlea. All these parts have air, not fluid. All right. So then you guys could just put descriptions in there. More activities that we're not really capable of doing, but you could read through them. Oh, this is a good table that kind of describes the difference between what's going on with semicircular canals. Remember, semicircular canals is more for rotational equilibrium. It gives you information if you're spinning, where the utricle and saccule is more for gravitational equilibrium, keeping you balanced, letting you know if you're accelerating, decelerating, or changing if you're moving up and down. All right. So you could do these activities if you like. Obviously, not going to be a requirement. And that's it. So look for the eyeball dissection. And I'm actually going to put another video that explains the ear model. When we do the test, it's going to be mostly eyeball, cow's eye dissection, naming the parts, labeling the parts, and identifying the parts of the ear on the models. All right. Good luck.